Welcome again. Today we return to the mouse mesocosm, and it's been 6 months. Today it's April 4th, 2015. In our last video on October 3rd, 2014, we set a timeline of 6 months to return to the mouse mesocosm to find out how the population might have changed in terms of the balance or the equilibrium of the various genotypes. Recalling that the albino genotype, which is Q squared in the Hardy-Weinberg equation, can be determined by looking at the phenotypes or the number of albinos in the population. So let's go into our mesocosm and first count the total population and then determine the number of albinos, and then we'll go to the blackboard and do some calculations. So let's count the little ones first. And now, let's count the number of albino mice in the adult and juvenile population. So let's move now to the blackboard and recall the two Hardy-Weinberg equations. 1 p plus q equals 1, which means that for two alleles in a population, the frequency of each allele adds up to the total frequency of all alleles in that population. And given this situation, and given the factors that must hold for Hardy-Weinberg equilibrium, such as random mating, no immigration or emigration, no natural selection, and a large enough population, given these factors, then the genotype p squared, which is the homozygous for the dominant allele, and the heterozygous 2pq, and the homozygous for the recessive q squared, all of these genotypic frequencies will add up to 1. And then given these two equations, it is possible to go into the mouse mesocosm and count the number of individuals that actually comprise q squared, and count the total population, which would represent 1. Given this, we were able to say that there were 10 albino mice out of 31 in the population. Therefore, q squared would come to 0.32. And given that q squared is 0.32, it means that q would be the square root of 0.32, which is 0.57. Given the fact that p plus q equals 1, if q is equal to 0.57, then p must be equal to 0.43. And this is where we begun the study on October 3rd, 2014. And we returned today, 6 months later, to do the same calculations, finding that there were 23 albino mice out of a population of 53 mice. During this time, there was certainly a lot of new births, for mice take just 5 days to go into estrus and just 21 days for gestation. There were also deaths and births, but it is clear from this number 53 that natality exceeded mortality, and there was no immigration or emigration. And from my qualitative observations, there was no evidence of sexual selection, and mating definitely appeared to be random. Given 23 out of 53, this time the frequency of the albino allele Q comes to 0.66, and the frequency of the dominant allele, the one that provides pigmentation, is 0.34. The question to ask is, how significant are these changes? A change in allele frequency of the recessive allele from 57% or 0.57 to 66%, and for the dominant allele from 43 to 34%. To determine whether this change is statistically significant, we employ the chi-squared test. Converting the frequencies into percentage to make the test valid, we also note that because there are two categories of data, that there is 1 degree of freedom, the output from the test generates a p-value which is slightly greater than 0.05, and in accordance with standard scientific protocol, it means that we should consider reasons to explain this value of 0.06, for it suggests that the variation between the two samples may be associated with something more than just pure chance. There is a significant difference in the frequencies of each allele over the 6-month period, and this suggests that one or more of the following might be true. The population was too small, and there might have been genetic drift, or there might have been some kind of sexual selection. I searched for this article from Levine, 1958. And here, Levine cites Urich, 1938, who found that within his strain of albino mice, the social hierarchy in some groups of mice was so rigid that only the dominant male was seen to mount the female. Levine's study goes on to present data that provides evidence that sexual selection may be a genuine phenomenon in mice. And although no such observations were made by me, it is an important issue to reflect on carefully and to aim to collect better data on this area in the future. Another factor could be that the population was simply too small, and then there was genetic drift. Genetic drift, or allelic drift, is the change in the frequency of a gene variant or an allele in a population due to random sampling of organisms. The alleles in the offspring are a sample of those in the parents, and chance has a role in determining whether a given individual survives or reproduces. Put simply, when a population is small, some of the genes can just kind of drift out of the population because the individuals that carry those genes simply do not reproduce. And when the population is small, there's a greater and greater chance of this kind of genetic drift happening. Take, for example, the spotted allele which existed in this mouse mesocosm in January 2014, but by July 2014, the allele was not seen in the population anymore. It was lost through genetic drift. The importance of genetic drift in evolution is something that is still being debated by biologists, and here you can see the views of Fisher and Kimura on this issue. What are the issues to consider for the future? 1. There is a possibility of sexual selection, and it is important to make regular observations of behavior and to mark all males, and to keep notes or make videos to document behavior. The second thing is to work with a larger population and a larger mesocosm to avoid genetic drift and to reduce competition or stress from limited space.